It's an honor to be here today to share some of our recent findings on a topic that continues to challenge and inspire the neuroscience community. It's a Parkinson disease. My name is Ouaki Hajar and my talk is titled Exploring Neuronal Changes and Neurobehavioral Response in the Parkinson's Disease Mouse Model. Before I dive into the detail, let me give you a brief overview of today's presentation. I've organized this talk into four main parts: introduction, where I briefly explain the background and significance of the study; materials and methods outline how we developed and assessed the Parkinson's mouse model; results and discussion, where I will present the key findings and what they mean in broader context of Parkinson's disease research; and finally, a conclusion. Parkinson's disease is the second most prevalent neurodegenerative disorder after Alzheimer's disease among the elderly, affecting approximately 2 to 3% of individuals over the age of 65 worldwide. As of 2020, it is estimated that 9.4 million people globally were living with Parkinson's disease. The disease shows a slightly higher prevalence in men than women. Parkinson's disease was first described by James Parkinson in his book An Essay on the Shaken Palsy, and he described very well all the symptoms developing in human patients, which are recent tremor, bradykinesia, hypomimia, postural instability, and muscular rigidity. So let me briefly explain each of this. Recent tremor is typically worsened by muscle relaxation or psychological stress and disappear during voluntary movements or while sleeping. As we can see in this video, this patient demonstrates predominantly left-sided tremor at rest affecting hand and feet. The tremor in the hands involves both fingers and wrists. It is most prominent when the hands are at rest but improves somewhat during action. Bradykinesia. So as we can see in this video, repetitive motor activity results in a progressive or persistent reduction in the range of motion, hypokinesia, and initiating movements becomes increasingly difficult. It's akinesia. So this bradykinesia can lead to a reduction in facial expressiveness and a decrease in other forms of nonverbal communication, such as gestures, is also common. Postular instability, which cause a balance problem and can lead to fall. So in this video, this patient recovers spontaneously with only one step backward, and the second patient who has more advanced Parkinson's disease and need to be called to prevent falling. Muscular rigidity, characterized by increased muscle tone, resistance to movement, and can reduce the range of motion. Parkinson's disease is associated with a wide range of non-motor symptoms like mood disorders, cognitive impairments, autonomic dysfunctions. These symptoms often precede the onset of the motor signs. Parkinson's disease is caused by the loss of dopaminergic neuron in the brain. These neurons are mainly found in the substantia nigra, a part of the brain that controls movements. As dopamine-producing neurons die, dopamine level decreases, and dopamine is a person for coordination, smooth and controlled movements. Without it, motor symptoms like tremors, rigidity, and bradykinesia appear. Another hallmark of Parkinson's disease is the presence of a Lewy body. These are abnormal clumps of a protein called alpha-synuclein that form inside neurons. Lewy body are found in the dopaminergic neuron of the substantia nigra and other areas of the brain. They disrupt normal cell function and contribute to neuronal death. Several factors may contribute to the degeneration seen in Parkinson's disease. We have age. The risk of Parkinson's disease increases with age. Most cases occur in people over 60. Men are more likely to develop Parkinson's disease than women. Some genetic mutation can increase the risk. And we have cranial trauma or head injuries, especially repeated trauma may increase the risk. And finally, we have environmental factor. Exposure to pesticide and toxin has been linked to a higher risk of Parkinson's disease. The heterogeneous of etiology of Parkinson's disease necessitates the development of prevalent animal models to study the disease mechanisms. The study aims to develop a subacute mirror model of Parkinson's disease induced by reserpine in order to assess the motor, behavioral, and biochemical alteration associated with the pathology. To achieve this, we used a combination of experimental technique designed to induce Parkinson's-like symptoms in the mice and assess their motor and behavioral function. We induce Parkinson's-like symptom using reserpine is a drug that depletes dopamine and other neurotransmitters by inhibiting the vesicle monoamine transporter VMAT2. This transporter normally packages dopamine into vesicle for release through synapse, and by blocking VMAT2, reserpine prevents dopamine from being stored and released, leading to depletion of dopamine in the brain, which in turn causes a neurotoxicity and neuronal death. In this study, we worked with two groups of mice, control group and Parkinson's disease model group induced by reserpine. This mice in model group received reserpine every 48 hours for 7 days. On the seventh day, motor coordination was assessed using the Rotary test, and locomotor activity, mood state, and cognitive function were evaluated using the open field test. Afterward, the mice brain were collected for chemical analysis to examine the neurochemical changes associated with the disease. For the statistical analysis, we used the Jamovi software. The comparison between the two groups was performed using student t-test. Now that we've discussed the method, let's move on the results of our study. We observed a significant decrease in locomotor activity in reserpine-injected group, as shown by a reduced number of squares covered in the open field test. Additionally, motor coordination was impaired with a decreased latency time in Rotary test for the reserpine group compared to the control group. The alteration of the emotional state was assessed by analyzing the time spent in the central and peripheral zone during the open field test. The reserpine mice spent significantly more time in peripheral zone compared to the control group. This suggests an increase in anxious behavior in the reserpine group. Cognitive function was assessed using the novel object recognition. The mice that received the reserpine spent significantly more time in front of familiar objects than new objects, indicating a cognitive impairment, and this suggests a particular deficit in short-term recognition memory in the reserpine-treated mice. At biochemical level, a disruption of antioxidant system and significant increase in oxidative stress marker were observed in the animal model of Parkinson's disease compared to a control group. These results therefore support the hypothesis that oxidative stress plays a central role in the pathophysiology of the disease, contributing to neuroinflammation, mitochondrial damage, and neuronal apoptosis. In conclusion, we found that repeated administration of reserpine effectively replicates both motor and non-motor symptoms of Parkinson's disease. These behavioral alterations are associated with increased oxidative stress in various brain regions, supporting the validity of this model as a valuable tool for preclinical research. Further investigations are needed to explore potential progressive alteration in dopaminergic system and other neurotransmitter pathway in this Parkinson's disease model. Thank you for your attention.